Thank you, David. I acknowledge my co-author, Dr. Catherine Bolger, and I also thank the organizing committee of Crop Updates to invite me to talk on Lyme effects on control of annual ryegrass and wild radish in low pH soil. Before I start, I want to acknowledge GRDC for funding this project and REUWA for research collaboration, and my colleagues Bob Sedge, Chris Roberts, and Deb Nichols on technical assistance. Chris Casey has been always been associated to this in providing technical advice, and our DAFWA research station at Geraldton, Meriden, and Wongan Hills, Biocrop Science and Labor Group. Now, why did we undertake this study? Anecdotal observation indicated that the weeds are apparently hard to be killed in acid soil, and application of lime make it a bit easier to control the weeds. In other words, the efficacy of herbicide increases as compared to the acid soil. Now, Gracie and Andrew, and you have seen the morning if you are here, you have seen that lime reduced ryegrass and radish impact in a long-term trial at Calabrian. So we asked the question, what is going on and what lime can do on weeds to control the weeds better. The first question is, what does lime do in soil and plants? Low pH soil means high soluble aluminum and manganese, and lime releases calcium and magnesium, and it reverses soil acidity, that means increases soil pH, and also reduces soluble aluminum and manganese. Now, the big question is, what does calcium do in plants and soils? It participates in uptake of other nutrients and promotes plant cell elongation, as it is an essential part of the cell wall of plant, and it participates in the enzymatic and hormonal processes, and it protects the plant against heat stress by regulating the stomatal opening and closing. I think from herbicide absorption and translocation point of view, this uptake of nutrients and enzymatic processes and the stomatal opening and closing are very important. So we thought, whether these changes is helping in the better weed control. So we aimed at examining the impact of lime and herbicide on the control of annual ryegrass and wild radish in low pH soil, and the hypothesis was application of lime should improve weed control and increase crop grain yield in a low pH soil. Okay, so what we did is we applied lime in the main plots at 4 rates in 2010 before sowing the crop. So the application was not easy. We spread the lime as precisely as possible by hand and without passing or spreading to the next door. And we had 320 plots, and we all spread all of them by hand in 4 locations. So in the subplots, we have 5 dates of herbicides. So different herbicide for different crops and weeds. So it was a split plot design with 4 applications, and the location was wild radish at Iradu and Wongan Hills, and annual ryegrass was Wongan Hills and Meriden. So the crop rotation for wild radish was wheat, lupin, barley starting from 2010, and we used velocity in 2010 and 11 and 2013, and in lupin crop we use simazine just one rate. In the herbicide treatment plots, in the untreated we didn't use simazine, and followed by a broadal multivariate mixture. Now, just to mention here, in 2010 we didn't have much weed in the plots, so we decided to repeat the crop and the herbicide in 2011. That's why you see the 2 wheat crop there. Now, for annual ryegrass, the rotation was same, but in 2010 and 11 we used Sakura at 5 different rates, and Boxer Gold in 2013, and in lupin crop in 2012 simazine as same as the other rotation. Now, the herbicides for annual ryegrass, we used Sakura at 5 different rates, because remember the subplot says herbicide. So, it is 0, 60, 90, 120 and 50, and that was in 2010 and 11. In 2013 we used Box Gold at 0, 1000, 1500, 2000 and 2500. So, we tried not to exceed the recommended level, but in case of this we had to go one step up. So, for wild radish we used velocity 0, 250, 350 and 500 mils and 670 mils, which is the maximum recommended rate. So, what we found, let us look at the soil pH first. Now, this is Iradu radish site. Now, the soil pH at the top 10 centimeter increased by almost 1 unit, and this is the data from 2013 and at the lime rate of 5 tons per hectare only. So, comparing no lime and 5 tons of lime, and at the 20 centimeter depth we had good increase at this site. So, this is obviously the recommended target pH of 4.8, this is above that, but as you go down to 30 centimeters subsoil layer, the pH increase was below that recommended target pH of 4.8. So, this means we still have soil acidity constraint at the subsoil layer in this site. When you look at Wangan Hills radish site, compare lime and 5 tons, we had the topsoil almost 1 unit increase of pH, but when you come to the subsoil layer of 10 to 20 and 20 to 30 centimeter, the increase was below that recommended target level of 4.8. That means we still have soil acidity constant there at both subsoil layers. At the ryegrass site at Wangan Hills, again topsoil we had 1 unit increase, even more, but when it comes to the subsoil layers, the constant still remains. So, the pH is well below 4.8 recommended target pH. So, this tells us that the even 4 years after the application or 3 years after application in the 4th crop, we still have subsoil constant, even though we applied lime up to 5 tons per hectare. So, this means the lime is slowly moving down the soil, but not enough at the subsoil layer as yet. So, we have constraint for the root growth of crops. So, let us look at the effect of lime on the initial weed density. This is the lime effect on wild radish at Iradu and Wangan Hills. So, this is the lime rate. This is the lime rate and radish plants per square meter, and this density is before application of post emergence herbicide or broadleaf herbicide application. But again, is worth mentioning here that this is probably a cumulative effect of all the years we have been doing the trials from 2010, 11, 12 and through 2013. So, for the first time in 2013, we have observed a reduction in the density of radish, and the overall reduction here is about 30% if we take the average of these three. Now, when I looked at the ryegrass and barley grass density, this is Wangan Hills and Merredin. At Wangan Hills, we had actually two grass species. We had the barley grass and the ryegrass. If you look at the ryegrass and barley grass reduction, there was a significant reduction with the increases in the lime red at Wangan Hills, and also at Merredin, where we had very relatively high density of ryegrass, and the reduction was nearly 60%. So, for the first time ever, we had significant reduction in the ryegrass density and barley grass density. We did not see this level of reduction in 2011 or 12. So, just to show you at Merredin site, this is no lime, no herbicide plot. The volume of ryegrass, you can see 276 gram as opposed to the barley crop biomass only about 41 gram, 31 gram. No lime, no herbicide. So, ryegrass has been growing very well, but not the barley crop. Let us look at the effect of herbicide on weed density. Herbicide has always been effective since 2011, when we had the first flash of weeds, but this is the result from 2013. If we compare the across the herbicide rate with the no herbicide, there was significant reduction, and reduction was about 60%. Now, if you look at the barley grass and ryegrass, there was also significant reduction as we increased the herbicide rate. So, herbicide was always effective in 2011, 12 and 13, whatever we used. And also in 2013, even though I plotted it against box gold, we need to keep in mind that this is the cumulative effect coming from 2010, 11 and through 2013. What is the effect of herbicide on the final weed control? So, this is the radish control. We had velocity in 2013, again this will be cumulative effect. So, the two sites at radish at Iradu and Wangan Hills, they are actually sitting on each other, and control level was very high as compared to no herbicide. So, it is 97 to 100% control. If you look at the grass herbicide at Merredin, the grass weed control improved with the increases in the box gold up to 2500. But when I look at the weed control at Wangan Hills, the control level at 1500 milliliter was equivalent to 2500 milliliter. So, it does not mean that we can reduce the herbicide, but we are getting this result. But overall control is 90%, but this is not to say that there was interaction between herbicide and lime, but this is just herbicide effect on the final control. Now, what is the effect on the grain yield? Let us look at lime effect. If you remember that we had very good control of radish at Iradu, but when I look at the yield due to lime, there was not much increase. At Wangan Hills, we had about 6% increase due to lime alone. So, there is a bit of complexity, bit of interaction, hidden interaction or confounding effect of herbicide and lime here. If you look at the ryegrass herbicide or ryegrass sites, lime effect at Merredin, there was not much increase, but again at Wangan Hills we had about 6 to 7% yield increase due to lime only. Herbicide effect on barley crop. Here we had spectacular effect at Iradu, where the wild radish density was very high. The yield of crop was more than double. At Wangan Hills, where the radish density was low, we had about 10% increase. Remember, the radish density here was very low. Now, at the grass sites at Meriden, the yield of barley crop was almost doubled or more than doubled as the herbicide rate increased. Compared to that at Wangan Hills, the yield increase was not that much, about 15–16% increase at the highest level of herbicide. Now, the one question I have been asked, did lime influence grain yield in absence of herbicide? So, to be able to answer that question, I just pulled out all the yield from no herbicide treatments and across the lime rate and compared the lime rate, and there I found that in absence of any herbicide, when I used lime, and there was an increase of about 37%. So, it is variable, but up to 37%. This is only lime effect in absence of herbicide. But when you go to herbicide, then it becomes more complex. The other question I asked myself, and I was asked as well, did lime interact with herbicide to enhance weed control? Well, that is a very important critical question, and that is the assumption we started with. The plain answer is yes, no. We did not see any strong interaction between lime and herbicide. In other words, in presence of lime, did we see any increase in the herbicide efficacy? However, in couple of occasions observed bit of a relatively weak interaction between herbicide and lime, only in case of high density of wild radish. For example, this one is wild radish at Iradu, where we had very high density, and comparing no lime with lime, and at 250 mils of velocity, the radish control increased by about 18% in presence of lime. Now, similarly, in 2013, we observed about 6% increase in presence of 250 mils of velocity. But notice that when we increase the rate of velocity, that differences have diminished. So, we can see the difference at higher level, higher rate of velocity. So, with that I conclude that lime increased soil pH, but the soil pH of the subsoil layers was still below the recommended target pH level even after 3 years of application. And lime reduced radish and grass weed density in all sites in 2013, but a small increase occurred in crop yield at 2 sites only. Herbicide was always effective on weeds and increased crop yield. However, a strong interaction of lime and herbicide was not evident. So, this results probably telling me the same story. It takes time to get the benefit from lime application, because lime takes time to move into the subsoils. So, for this kind of plot, we may have to wait longer time. As the previous speakers, especially Chris Casey, was saying that the lime has to be applied at certain interval, and then it can be incorporated by cultivation to get the immediate result. So because we didn't do the cultivation in this one, the incorporation was only just by normal sowing. We may have to wait to get the benefit for this, but we have a limit on the time of the project. We may not be able to go beyond next year. So with that, thank you very much. I'll be happy to answer a question if you have any.
What is it that's actually driving that good result with the wild radish? I think it was at the Eridu site, your higher lime rates, you had fewer wild radish in the plots in 2013. What is it that's causing that? Is it the extra competition from the crop, or is it, what is it? Or is it something to do with the, the germination of the radish in the changed pH?
Yeah, that's a good question. I have been asking myself and discussing with other friends and colleagues as well. So what happened in 2010, why we are getting this significant reduction in wild radish? There are some speculation. I don't have exact answer. So the first thing is lime causing something to radish? As a result, the radish seed bank has gone down. Well, my observation in 2011-12, there was a bit of higher emergence in the lime plot, but the results were not significant. So it can be a cumulative effect of more emergence over 3 years, and we got this significant difference only in 2013. That can be one explanation. The other explanation is it could be possible that the seeds are in the soil. They didn't get the right window to germinate in 2013. So if that is the case, what do we conclude with that? But again, that is again speculation only. So we need to do the soil sample and test whether the seeds are there or they are dead. Now the other thing is whether calcium carbonate or egg lime has got some magnesium carbonate as well. Whether these two can do anything to wild radish germination. So I tried to do some literature search. There is some information calcium bicarbonate can induce dormancy to some plant species, seed dormancy, but calcium carbonate does not induce dormancy to any. I didn't see any literature, but magnesium carbonate can reduce the germination of seed. So, but magnesium carbonate, if I am correct, you can correct me if I am wrong, only about 10% in egg lime magnesium carbonate. So if it is, the figure is correct, then it doesn't have loss of magnesium carbonate in there to cause that kind of change. So my guess is the other explanation could be like that in a year like 2013, we had a bit dry period in June, July, whether that has reduced the emergence, that could be possible as well. So it's again subject to investigation whether the seed is there or it is the cumulative effect of emergence over the last 3 years.